Okay, so we'll start the session now. Hmm. Someone has messaged Ajay. Exam date is near Deepavali. Okay, so we cannot help it out. If your exam date is near Diwali, then you have to uh, write it. That is not in my hand. If you have any problem, you can talk to the NPTEL people. They might help you. But as of now, whatever date has been decided, it has to be done on that date only. Okay, so a very good evening, everybody. So today we are going to discuss questions which are related to the week three class. So in week three class, it is mostly about um, differential gene expression and along with some basic concepts of genetics. Okay, so as we've been studying, we studied in our last class also that although all genes are present in all the cells of the body, okay, we talked about genome equivalence, which says that all the genes are present, all the chromosomes are present in all the cells of the body, most of the cells of the body. Then how is it possible that some genes are expressed in some particular tissue type and they are not expressed in another tissue type? For example, if the gene which is responsible for your eye color, it is not expressed in your liver or lungs or anywhere else, it is specifically expressed in your eyes. So how is this possible? It is possible because of the differential expression of genes. So now the differential expression of gene is controlled by many methods, and one of the method that has been dealt this week, if you have gone through the lectures, is alternative splicing. That means that the same mRNA can be cleaved in more than two ways, which leads to different mRNA products and which will make different proteins. So, just a minute. Hmm. So you can see this is your differential splicing, or we can call it as alternative splicing. So in what happens in an alternative splicing? In alternative splicing, you can see this is the mRNA. These are the introns. The gray color ones are the introns, and the colored ones are the exons. So your introns are the non-coding regions, and your exons are the coding regions, right? So you can say that the combination in which these exons join, you can see here the splicing has occurred in such a way that your exons 1, 3, and 5 have joined over here. Whereas here, splicing has occurred in a way that exons 1, 2, 4, 5 has joined. So, the difference in the joining of the exons depends on the kind of splicing that is happening, and which determines the different mRNAs which are produced, ultimately leads to the different protein production. Just a minute, I think someone has joined the call. Yeah, fine. So this is your differential splicing, which can lead to a differential expression of gene. Other thing is RNA selection. What is the meaning of RNA selection? We all know that the mRNA is produced inside the nucleus. Okay, just a minute. Hmm. So your mRNA is produced inside the nucleus, and after its production, it goes to the cytoplasm where it is taken up for protein synthesis, that is translation. So your transcription and replication, it occurs in the nucleus, right? And your translation, it occurs in the cytoplasm. So now the different RNA selection can also lead to differential gene expression. How come? That in certain cell type, you can see that C, D, and E, out of all the RNAs, C, D, and E RNA were taken out to be expressed and translated into proteins. Whereas in cell type 2, you can see that A, B, and C RNA were taken out to the cytoplasm to be expressed into proteins. So the expression also depends upon the RNA selection. That suppose in a particular cell type 1, let's say in cell type 1 only, your gene B, sorry, mRNA B is not needed. It is needed only specifically in cell type 2. So the easiest way out is that just keep this mRNA inside the nucleus, because once it will go to the cytoplasm, only then it will form the protein, only then it will get expressed. So this is a way of differential gene expression through RNA selection and differential splicing. Okay, so with this little background, now we'll start to the questions. No, you cannot call it as a pre or pro RNA, because you cannot call it as a pre RNA. That is a different thing. Okay, so now first question is, so like we always do, this is the question. I'll just read out the question and then I give you 2 minutes. You have to think of the answer and then you have to message it. I mean, write your answer in the chat box. Okay. So don't hurry up. Like, let others also finish. So don't unmute yourself and instantly start speaking the answer, because we are in a class. We have to take everybody together. So if you just read the question, if you know the answer, then just write it in the chat box. Okay, so the first question is, function of RISC is to find the complementary strand and cleave the RNA, to inactivate the RNAs and protect the RNA from the risk of cleaving, to find the complementary strand and cleave the DNA, and to inactivate the DNAs and protect the DNA from the risk of cleaving. So I'll give you 2 minutes, try to think and answer, then I'll switch the screen. One more minute, I'll switch the screen then. Okay, then now I'm switching the screen. Hmm. Let's see what you have answered. Okay. So Ajay and Nazreen has answered. So Ajay has answered A, that is to find the complementary strand and cleave the RNA. And Nazreen has also answered A. So it is correct. So the function of RISC is to find the complementary strand and cleave the RNA. So this is just a general overview of the miRNA biogenesis. Okay. Uh, we'll see here. Okay, just let it be like this only. Fine. See here that this is your miRNA gene. So this is actually how your miRNAs are formed. So it is miRNA biogenesis. So this miRNA gene, when after transcription, it forms your pre-miRNA. Okay. Now this pre-miRNA is cleaved by drosha to form the pre-miRNA. So always remember pre, P-R-I, pre-miRNA is converted into pre-miRNA. Okay. And this conversion takes place with the help of the enzyme drosha. So drosha cleaves pre-miRNA into PRI pre-miRNA into PRE pre-miRNA. And then this pre-miRNA, so all this is happening inside the nucleus. Okay, so as we all know that transcription and the post-transcriptional modifications, if there are any, they happens inside the nucleus, and after the trans modifications or whatever processing it has to undergo, after that only your miRNA or your RNA, it comes out of the nucleus for translation. But here the concept is not about translation because it is a microRNA, but to assign its function it has to come out of the nucleus. So now this miRNA, it comes out of the nucleus. This is your pre-miRNA, and this pre-miRNA is acted upon by dicer, and then you can see that this kind of duplex is formed. miRNA duplex is formed, and then it goes to the risk compartment, and from here it performs its function, which is either translational repression or mRNA degradation. Now one important thing is that this pre-miRNA, after the processing by dicer, it forms two different strands. Okay, so what are those different strands? The first strand, um you can see here, the pre-miRNA is exported to the cytoplasm and undergoes cytoplasmic processing to produce mature miRNA duplex. So one is your normal strand, one is your passenger strand. So asterisk represents passenger strand. Okay, so you can see that your mature miRNA, it is it that a mature miRNA is actually responsible for your miRNA degradation or translational repression. This passenger strand is degraded. Sorry, passenger strand is responsible for degradation. But miRNA, which is mature miRNA, it is responsible for repression and degradation both. Now, what is the meaning of mRNA degradation and translational repression? So, this mature miRNA which has formed, if it is perfectly complementary to the mRNA, it will degrade the mRNA. Okay, so inhibition of the transcription process. What is the function of microRNA? These are nothing but they causes the repression of expression. They inhibit the expression of genes. Okay, microRNAs, they inhibit the expression of a particular gene. Now there can be two ways to inhibit this expression. First way is through the degradation of mRNA, and second way is translational repression. That means mRNA is degraded. Now there is no chance that mRNA can go for the next step, that is translation. Secondly, what it can do, that okay, mRNA is synthesized, but what is the next step of mRNA? It is your protein synthesis, that is translation. So this translation process is inhibited. So in either of the ways, the ultimate goal, that is the production of the protein and the expression of the gene, that is getting stopped. So either by directly degrading your mRNA or by repressing your translation process, this miRNA blocks the expression of that particular gene. So this is how, it's a very general and basic overview of your mRNA biogenesis. Okay. Okay. Now next question is, it's similar to the one that we have dealt just now. Hmm. Huh. PRI pre-miRNA is converted to pre-miRNA, that is PRE. Just pay attention to this. By the action of dicer, risk, drosha, or none of the above. 2 minutes and then I'll switch the screen. Okay, now I'll switch the screen. It's an easy one. Hmm. Hmm. Okay, so I think Nazreen, Ajay, and Chauvin all have answered it correctly. That is C. We have just discussed it is Drosha. Okay, so these were the simple questions. Now we'll come to some of the. Okay. Next question is bicoid mRNA is transported to which area and motor protein responsible for the transport. So there is nothing to explain here. It's just like a basic question that the information need to be memorized. But I'll give you 2 minutes anyways. Try to answer it, and then we'll come to the later part of the questions, which are actually the main ones. Okay. Now I'll switch the screen. Let's see. Hmm. So Ajay has answered C. Chauvin has answered C. Okay. Let's see what is the C is anterior dynein. And yes, it's the correct answer. Okay. Fine. Now, next question from here. These are the actual questions that I wanted to discuss today because I think they are the basics of genetics. They will form the basics of genetics. So try to answer this question. These are little not these are very easy only. And I would request I just a minute. Is there any option to send an image here? Can you send an image in the chat? Is it possible? Because I wanted to see how you have solved the question, not just the answer. Okay, you can type a little bit. So for these genetics questions, I would request not just you answer the like A, B, and C, but also like a little bit detail about how you have solved it. Because then only I'll get to know whether you are doing it correctly or not. Because there is no option where you can send an image. Otherwise, you could have just clicked the picture of where you have solved. So just keep and copy and pen with you because from now all the questions are like genetics based only. We have to solve it. Okay. Now comes the interesting part. Okay. So your first question is a man with sickle cell anemia married a healthy woman without sickle cell anemia. What is the probability of their child suffering from sickle cell anemia? Okay. This is the question. I'll give you 5 minutes. I'll just switch the screen like this. I think it is visible enough. I hope it is visible enough. Yeah, okay. Hmm. This is the question. It's 6:20 right now. At 6:25, I'll switch the screen. 5 minutes is more than enough for this question. Try to solve it. Okay. I'm just saying. So Ajay has answered. Okay. Nazreen, no. Okay. Okay, I'll just give you one more minute and then explain this question to you. So I understand what Ajay you are trying to say. 100% trade, 0% disease. Okay. Okay. Fine. I think Ajay you have answered it correctly. Okay. Okay, I'll explain to you now. So a man with sickle. So everybody please pay attention. Even if you couldn't solve, it's fine. But at least pay attention to now what I'm saying. Okay. Nazreen, the children will be carriers only. Okay. Fine. Now look here. Hmm. Just a minute. Hmm. A man with sickle cell anemia married a healthy woman without sickle cell anemia. So, before answering this question, first thing that we should know is what kind of a disease does sickle cell anemia is. So, can anybody tell me what kind of disease is this? Sickle cell anemia on the basis of genetics. Autosomal recessive, yes. So Ajay has answered it correctly that sickle cell anemia is an autosomal recessive disease. What is the meaning of autosomal recessive disease? That means this disease is not gender specific, that it will be transferred from fathers only, fathers to their daughters or to their grandchildren, or it will be transferred from mothers to their son. No, it is nothing like that. It is an autosomal disease. It is not a sex linked disease. First thing is clear. Then recessive. Recessive means only when the two recessive alleles are present, then only this disease will show its phenotype. Otherwise people will be just carriers. Okay, so this is clear that sickle cell. Okay, just a minute. Hmm. Sickle cell anemia. It is an autosomal recessive disease. Now the question says a man with sickle cell anemia. Man with sickle cell anemia. Now the man has the disease. So if let's say if sickle cell anemia is coded by A, so then man is small a, because this is the dominant one. Let's say this is the recessive one. Okay, and man has the disease. So this is small a, married a healthy woman without sickle cell anemia. Now woman is healthy without sickle cell anemia. So they have not mentioned it correctly, I mean clearly, that whether the woman is a carrier or not. So we'll consider that woman is not the carrier, she is healthy. So if woman is healthy, that means her genes, I mean her alleles are AA. Now with the help of Punnett square, let us try to solve this. This is AA, this is AA, 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 AA. So you can see that this, since it is an autosomal recessive disease, so it can only be caused when both the alleles are recessive. And here you can see that none of the alleles are recessive, they all are the carriers. So the correct answer would be 0%. So none of the child would suffer from sickle cell anemia. Now just a small change. What if the question was like this, that man has sickle cell anemia, okay, and woman is a carrier? Then what would happen? Now tell me. if I am specifying that woman is a carrier? Ajay, you have raised your hand. Yes. Okay. Yes, 50-50%. Now, if the woman is carrier, that means specifically they have mentioned, specifically it is mentioned. Yes, Ajay, please tell me. Yes, you can
Madam,
speak.
the framing of question is totally wrong.
Yeah, I mean, they have not,
They
they
should
have,
have mentioned first male and female. Why they are mentioning man and woman? I don't know. Second, sickle cell anemia. Anemia is a temporary condition, symptomatic condition. They
I
should have
think,
mentioned sickle cell disease or trait.
see, these are the questions that I have not framed, okay? These are the questions that NPTEL has
No, that's
provided
what I'm
me.
saying. Even madam, spelling mistake, they had given question wrong. Means chimara, chimera, whatever that. Just
I
one
think
spelling.
that's fine. See, our objective is to understand the concept. If we are able to understand the concept, that's enough for us. Whatever mistakes they have done, let there be. We cannot change it now. These are the questions that they have provided and they have only framed. So, anyways, it's good that you could understand what the question is trying to say.
But
Okay,
it is
so
percolating in a wrong way. No? Anemia is a temporary condition. Patient cannot be, a man cannot be with anemia. He,
okay,
with disease or trait. Sickle cell anemia is a group of disease in which both are included, trait and disease.
okay. So I
A
think
may,
I think that's really good of you, Ajay, that you are trying to uh, tell even more about this disease. But let us try to focus on the question right now because we have a lot many questions to cover. So, okay. So what I was trying to say here is that if woman is a carrier, then what will happen? So if woman is a carrier, that means it is specifically that it is AA because then only she will be a carrier. So now what will happen? This is AA and this is AA. So it would be AA here. That means 50% are normal and 50% are suffering and 50% child would be normal. The probability. Okay. Now we'll come to the next question. It's an easy one. 2 minutes for this question. A person having A blood group will have anti B antibodies, antigen A, antigen B, anti A antibodies. 2 minutes to answer this question, and then I'll switch the screen. It's an easy one. You should be able to answer. Okay, now I'll switch the screen. Hmm. Question is a person having A blood group will have. Okay. See what you have answered. Okay. Hmm. So Ajay has answered antigen A and antibody B. Anti B. Okay. Correct. So Ajay has answered it very correctly. So just a minute, I'll just show you if you haven't gone through the lectures, which you should have at, but I don't know it's in which class. Is it in second class? Okay. Hmm. I think it is in the first class only. I don't know where it is. Okay, let it be. I'll just explain by myself. So, for sex determination, if a person has A blood group, that what does it means? That in its body, in his body, A antigen is present. Okay. So, if you have your blood group as A, then which antibody would be there? Anti B antibody would be there. If you have a B blood group, then which antibody would be there? Anti A antibody would be there. If you have A B both blood group, then what will happen? Can anybody tell me? If somebody has an AB blood group, then what will happen? If you have AB blood group, what kind of antibodies would be there if somebody has an AB blood group? Quickly. Meanwhile, I'll search where is it. Anyways, let us see if you have answered what I'm trying to ask. No antibody and both antigen. Yes. So if somebody has AB blood group, that means they do not have any antibodies and they have antigens for both A and B. And if somebody has O blood group, that means they have antibodies for anti A and anti B both, and they have no antigen. So this is just a very basic question. This I think you have been studying since standard 12th. But even if you do not know, just remember the blood group signifies the antigen that is present in your blood. So if I say blood group A, that means you have A antigen. If I say blood group B, then you have B antigen. If I say blood group AB, then you have AB antigen. If I say blood group O, that means you have no antigen. So if you have A antigen, so what would be the antibody? Anti B. For B, anti A. For AB, nothing. And for O, both anti A and anti B. Okay, like this you can remember. Okay, now again comes a question. I'll give you 5 minutes for this question. Okay, it's a very interesting question actually. A recessive allele causing a particular disease show independent assortment with the ABO blood group. The disease only affects individuals of blood group O. A normal man with a blood group A and a normal woman with a blood group B already have a child with disease. What is the probability of the couple having a second child with the disease? Your options are 1 by 2, 1 by 4, 1 by 8, and 1 by 60. Okay. Now, I'll give you 5 minutes. Okay. It's 6:36 or 6:35. So, at 6:40 approximately, I'll switch the screen. Read the question carefully. Try to understand what they are asking and then solve it, and just a little bit if you can write just like you wrote earlier also, just like Ajay has written very correctly. So try to write something in the chat box also. Okay. And even if you don't know, doesn't it's not it doesn't matter. I'm anyways going to explain the question. Think there are two things that are being asked in this question. One hint I'm giving you. There are two separate things that you have to solve. This is one hint. It's a major hint if you can take it. Okay. First line is very important. A recessive allele causing a particular disease. Okay? It is a recessive allele also, and it causes the disease also. Two separate things. Two more minutes. One more minute left. Okay. Shall I switch the screen now? Okay. So Ajay has answered 1 is to 16. Others, Nasreen, Shoven, Shubhamoy. Can you think what can be the answer? Any guess, like any basic concept also if you know? Okay, 1 is to 16. Okay, good. Yes. So the correct answer is 1 is to 16. Now I will explain it how. Okay. Just a minute. I'm sorry if I'm actually eating in between because I haven't eaten anything today. This is my first meal of the day. Hmm. So now you see the question. A recessive allele causing a particular disease show independent assortment with ABO blood group. Okay. The disease only affects individual of blood group O. Now this is important. So in this question, just a minute. Hmm. So in this question, there are two things. Two independent things are there. First is inheritance of the ABO blood group that we have to keep in mind. Second is the inheritance of the disease pattern. Okay. Now, a recessive allele causing a particular disease shows independent assortment with the ABO blood group. So, disease only affects individuals of blood group O. That means, uh, that it has to be IO, only then the disease can show its phenotype or its effect. Okay. So, a normal man with a blood group O and a normal woman with a blood group B already have a child with disease. So, if you have a blood group A, then what can be the genotype? It can be either IA, or it can be IAIO. Similarly, for blood group B, it can be either IB, or it can be IBIO. Now, we know that the disease is caused only by blood group O. That means it has to be present in a recessive condition. So for that, it has to be I A I O and IBIO. Otherwise, how would O would come? Okay, so that means it is like this: IAIO and IBIO. So it is IAIB, IBIO, IAIO, IOIO. So here you can see that this is the condition in which disease can be caused. So what is the probability? The probability is 1 by 4. So from here we can say the probability is 1 by 4. Now this is one thing. Another thing is um, inheritance of the disease. Now this disease inheritance, suppose if this is your father, so since the disease can be affected, let's say disease D is the normal and this D is the diseased one. Okay, this allele is the normal one, it will produce a normal phenotype, and this allele for the disease, it will produce a diseased phenotype. So father is DD, mother is also DD. So from here, if you do the Punnett square, what would you get? DD, 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 and DD. So from here, for the inheritance of the disease, the probability is again 1 by 4. Now, this is a classic example of independent, when you have two independent traits or independent things, then what you have to do? You have to multiply. So 1 is to 4 into 1 is to 4, which is equals to 1 is to 16. So it would be 1 is to 16. Okay. Okay. Your next question is, so after we'll solve all the questions. Okay, not okay. Can you answer this question? Because just answer this question. I want to see what you answers, and just let me know because I am a little confused about this question. So let us see if my what I'm thinking is correct or I am missing some point. Okay. So for a cross between the genotype this and this, what is the probability of getting an offspring with the recessive phenotype of all four traits? Okay. These are your options: 0, 1 by 2, 1 by 4, 1 by 8, and 1 by 16. Now try to answer this and then we'll discuss it. Okay. You have 5 minutes to answer this question. Answer, think, answer, and just put little bit information in the chat box. Okay, think and try the question. Okay, let us see what you have answered. 646. R is. Hmm. Hmm. Can you explain how you have hmm. So how you can say this? So it's a very easy question actually. Why? Because here they are asking what is the probability of getting an offspring with the recessive phenotype for all four traits. Now I am first telling you in the basic, like the the general way of solving this question. Like here it is zero, but in some cases it might be some fraction. So how you have to solve these questions? Okay. After that, we'll see ki why it is zero. Okay. See, first is um, so these independently we have to solve for each and every one. So you see, this is the cross. So first we'll solve for P gene, then we'll solve for Q genes, then we'll solve for R genes, then we'll solve for S gene, and then we'll multiply whatever the fractions or whatever the probability comes. Okay. Now for PP and PP. Oh, now, Ajay, I have a confusion. Can you tell me how you have solved this question by unmuting yourself? Can you hear me, Ajay?
Hello?
Haha, yes.
Madam, I had multiplied 1 by 4, 1 by 4, 1 by 4 three times. It falls to 1 by 64, and the
Hmm.
option is not here.
Actually, I think, see,
You are
you have
correct.
to.
Some fraction is there, but probability is 0%.
Fraction is
One
there,
of
probability. That is fine. See, if this is P and this is a small p. Okay, this is small p. So when I make Punnett square, 1 by 4 would come in. This is
actually,
recessive
madam,
one.
we make Punnett square by 1 by 4, 1 by 4. Actually, it is 1 by 16, 1 by 16, 1 by 16 three times.
Yes. So I don't
So,
know whether this is a little okay. We'll just keep this question aside because I don't know whether
so
this question is
it is chance is 1 upon 4096.
that it is the options are incorrect actually.
Uh,
Um, so just leave this question aside because I don't think this is the options are correct. Anyways, we'll first solve this question and then I'll come to how we have to solve this question like normally. But first we'll solve this question and we'll just remember this question. We'll discuss it in the next class if it is not possible in this class, because I have some doubts regarding this question. I don't think this question is the answers are correct. Okay, anyways, so now this is a very big question. It might take a while for you. Okay, but it's a very important question. Why it is important? Because this question is very important for your competitive uh, per se also quantitative competition exams point of view also. So because these kinds of questions are frequently asked to find the recombination frequency or to find the gene order. So the question says, determine the correct gene order and calculate the recombination frequency between P and R. So I want to give more time on this question because it's a big concept. So first you try to solve by yourself and just write little bit how you have solved it, and then I'll tell you how to actually solve this question. But first try yourself. If you have studied genetics, I think you will be able to answer this question. I'll give you 5 minutes for this question. It is 6:54. Okay. At 7 o'clock, I'll switch the screen. Okay. At 7 o'clock, I'll switch the screen. Read the question thoroughly and then answer. It will take a while for
पेन
me
है
to
क्या एक
explain.
पेन भी तो
Sorry, please repeat.
माइक पर वही
Okay. 7 o'clock, I'll switch the screen. See the values. Whatever values are given now, these values, these are actually the hint to solve the question. See these values and then try to solve the question. Okay. 3 more minutes left. 1 more minute left. Okay, I'm switching the screen. Hmm. Let us see. Hmm. Hmm. Yes, it is a 3 point desk cross. Okay, so Ajay, you have answered it correctly. So can you explain to the class how you have solved it? Okay, chalo. I'll only explain. Hmm. Now you see, this is an example of a 3 point test cross. Okay. Here we have to find the gene order. Now, first and foremost thing, whenever a question which is related to a 3 point test cross is given, what you have to do is that you have to observe the parental classes and the double crossover classes. Okay. So, what are the parental classes? Parental classes are the classes in which maximum uh, number of individuals are present. So, in this case, your 3900 and 3700 are the parental classes. Okay. And double crossover are the classes in which least number of individuals are present. So, these are the double crossover classes, and rest four are the single crossover classes. Is this clear? The one with the greatest number, they are the parental classes. Least number, double crossover, and the rest, they are single crossover. Now, okay, in the next slide, actually what it is, I have to see. I have to write it down in my copy somewhere so that I can write it on the. Hmm. Okay. So, one of your parental class is PP. No, first I will not. First I'll do it here. Okay. This is your parental class P P Q Q R R. Okay. And this is your another parental class I am writing over here. P P Q Q R R. Is it correct? P P Q Q R R. Okay. These are your parental class. Now let us see the recombinant classes. Sorry, double crossover classes. So these are the two double crossover classes. So I will write it here. You see, this is your double crossover class P P Q Q R R, and the next one is P P Q Q R R. Okay. So what is the gene order here? Can anybody tell me from this? Can anybody tell me what is the gene order? Gene order is, anybody?
You are
P. What is the gene order again? Can you say it again?
PRQ.
PRQ. Yes. Now why it is PRQ? Because just these are the double crossovers, and this is the parental type. You see here, this is exactly the same. This is exactly the same. Here the crossover has happened. Again, this is exactly the same. This is exactly the same. Here the crossover has happened. So the gene where which actually where the gene which shows crossover, that gene is present in the middle. This is the concept that you have to remember. So here you can see that in both the cases, because it's a double crossover, you can see that uh, in both the cases your R gene is getting uh, uh, crossover by between the I mean your parental generation and your recombinant generations. Okay, so from this you can say that R has to come in between. So it is PR Q or PRQ, or from this way QRP, that is fine. So from here you can you see any option PRQ? Yes, it is there. So from here I can say that for the first one it is PRQ. Okay, so like this you have to find the gene order. You have to see that at which position your at which gene your crossing over is taking place, and whatever gene is getting crossed over, that gene has to come at the middle. So here, since R is getting crossover, you can see here this is same, this is same, this is same, this is also same, but here it is uh like the other another it is like the other parental generation. Here also you can see this is also same, this is also same, but this is the of the other parental generation. It means crossing over has taken place. So your R is in the middle, so your gene order would be PRQ. Now this is the first thing. Now we have to calculate the recombination frequency between P and R. Okay, now let us in the next slide I will arrange this parental this thing parental genotype, but now in its correct order. So how it will be? Just a minute. Hmm. So how it will be? See, yes, I have written here. Now I am writing all the classes here. Okay. Since our gene order is, as we could understand, that our gene order is PRQ. So same, just try to match with the question as well. Same parental genotype. Now I am writing in that order. So for PP in first parent, it is PP. For R, it is RR. For Q, it is QQ. Okay, this is your parental one. For parental 2, okay, for parental 2, how it is? It is PP, RR, QQ. These are your parental. Okay, fine. Then your single crossovers. Now we'll take the single crossovers. For single crossovers, we'll start with each one 270, 300, then 80, then 810. So for 270, you can see it is P, P, R, R, Q, Q. Okay, so you can see from here crossover has occurred in QG. For your 300, it is P, P, R, R, and Q, Q. Okay, here also crossover has occurred in QG. For the other single crossover, which is 810, how is it? PP, R R Q Q. Okay. When we compare this with parental generation, where the crossover has occurred? At the PG. Next, we come to 850. It is PPRR and QQ. Okay. Now compare it with the parental generation, where your crossover has occurred at P. Now in double crossovers, where the crossover has occurred at R. See here, we have already discussed that in double crossovers, your crossover has occurred at R. That is why it was in between. Okay. Now we have to calculate the recombination frequency between P and R. So as we have to calculate the recombination frequency between P and R, we have to take into consideration all the recombinations that has happened in P and R. Okay. So what are the recombinations that has happened in P and R? Recombination, this is 810, which has happened in P, and plus 850, which has happened in P, and in R, what were the two recombinations? The double crossover, 50 and 80. So 50, sorry, 50 plus 80. Okay. So how much it will come? You can calculate. Then, so the general formula is the classes, identify the classes where recombination frequency has occurred upon total number of classes into 100. This is the formula. So the classes in which recombination has occurred between P and R. So we have to consider the recombinations of P also, and we have to consider the recombination of double also. So the what were the values of the individuals that has recombination in P? It was 850 plus 810, and recombination between R, it was, I mean, the double crossovers 50 and 80. And if you sum up, this will come as 10,000. You can just do the calculations, and when you calculate it, it will come approximately 17 point whatever percent, 9% or whatever it is. So like this it would come. Okay. So there are certain, I'll just again repeat it. So for in these 3 point test cross questions, if a table like this is given to you, your first thing would be to look at the numbers. Okay. So when you will look at the numbers, you will first identify the classes which are parental, and you will identify the classes which are double crossovers. The parental classes are the ones which has the maximum number of individuals. In this case, it is individual. And your double crossovers are the classes which has the least number or the lowest number of individuals. Okay. And the rest are your single crossover classes. Now we have identified parental and double crossover. Now second task is to compare your parental classes with your double crossover classes. Okay. So first is to identify the different classes, and second is to compare your parental class with your double crossover classes. So here you see, this is the parental one, PPQQRR, and when I compared it with double crossover class, I observed that there crossover has happened at R gene. Similarly, when I compared my parental class with the double crossover class, I observed that the recombination has occurred again in the R gene. So whichever gene shows the crossover, that gene is present in the middle because it is a 3 point test cross. Okay. So from here we can say that the gene order is PRQ. Fine. Now we have to find out the recombination frequency. So if this is your P and this is your R and this is your Q. So for finding out the recombination frequency, you have to find out the classes which shows that respective recombination divided by the total number. and into 100 because we are calculating frequency. So if I am talking about recombination frequency between P and R, that means I have to identify all those classes where recombination of P and recombination of R has happened. So if you compare, just like I showed here, if you compare, you can see that recombination of R has occurred in the double crossover classes. We already dealt with it. For P, 810 and 850 in this classes recombination has occurred. So then what we'll do? Simply 810 plus 850 plus 50 plus 80 upon total, which is 10,000 into 100, which will come out to be 17.9%. So like this, uh, questions of 3-point test cross can be solved. Okay, I hope it is clear to you. Hmm. Is it clear to everybody this question? Because it is an important concept which is frequently asked in your competitions. That's why I'm putting more stress. I uh, kept more stress on this. Yes, Ajay, you can ask.
Madam, how much time is given to solve this type of question?
Um, that
Because
even
I
I
think that
don't
is
know. I think
because it will.
you will have a complete set of questions, and it depends upon you that how much time you consume on these kinds of questions, because it would not be like that for each and every question you have a time limit. Suppose whatever, I don't know what is the exam pattern, how much time you have. So how much what is the time, like is it 3 hour exams or a 1 hour exam?
I think 3 hours, but
Okay,
questions
so like
are much. This-
yeah,
Since they are objective questions, hardly
I
one
think
minute on 1.5 minute is given to each question.
okay.
Usually,
Okay. So if that is the case, then you have to speed up, because these kinds of questions are also asked in your CSIR in part C. And in part C, mostly lengthy questions are only asked. If you want to pursue for PhDs, anybody, then these kinds of questions are only asked in your part C CSIR. So there also you have to be a little more like time management is important. So that's what I told you, that try to solve as many questions, and just as soon as you see these kinds of questions, immediately try to identify your parental and your double crossover class, and try to compare them. As soon as you can compare, it is easier for you now, because but always remember, when you have to calculate the recombination frequency between any two genes, if you start calculating recombinations frequency by randomly like this PQR, then it would be difficult for you. You won't be able to get the correct answer. So for calculating the recombination frequency, just rearrange the genotype into the correct gene order, and then only calculate the recombination frequency. Okay. Because if you start calculating by like this PQR, by considering this order PQR as given in the question, then it would be difficult for you to calculate. It will take more time. So try to first arrange the like here it was PP QQRR. I arranged it for comparing. I arranged it. I kept R in between. So for this comparison, that what are the single crossover classes where the recombination has been taken place, you should arrange it first in the correct order, and then only you should calculate the recombination frequency. This is an important point. Okay. Now we will come to the next question. It is interesting only. Okay, so for genes A, B, and C, the distance between the two extremes is 21 centimorgan, sorry, and the recombination frequency between C and B is 15%. Which of the following can be inferred? The order of the gene is ABC, and the distance between A and B is 6 centimorgan. The order of the gene is ABC, and the recombination frequency between A and C is 21%. The order of the genes is ACB, and the distance between A and C is 6 centimorgan. The order of the genes is ACB and the recombination frequency between A and B is 21%. The order of the genes is CAB and the recombination frequency between A and B is 6%. Or the order of the gene is BAC and the recombination frequency between B and A is 6%. Okay. So I'll give you 3 minutes, 3 to 4 minutes. Try to understand the question carefully and then try to answer. Okay. It's easy. If you could solve the previous one, you can definitely solve this one also. 3 more minutes and then I'll switch the screen. I want 100% participation. Anyways, there are so only few students who are coming to the live sessions. I don't know why. Either they know everything or they don't want to know anything. One more minute. Okay. Now I'm switching the screen. Hmm. So, okay. So A, B, C, and D. B. I don't know what you have answered. Okay, fine. Anybody else? Nasreen, Shubhamoy, Showan, or Showan. I'm sorry if I'm pronouncing your name incorrectly. Any other response from anybody else? No? Why? We just solved a question similar to this one only. Our previous question was very much based on a similar concept. That means you didn't understood the previous one also. Hmm? Okay, chalo, I'll explain it anyways. Okay, so the question says for genes A, B, and C, the distance between the two extremes is 21 centimorgan. Okay, so if I just write these genes A, B, and C at this point of time, I don't know the order, I'm just randomly writing it. Okay, now the distance between the two extremes, okay, this distance is 21 centimorgan. And the recombination frequency between C and B. C and B is 15%. So if the recombination frequency between C and B is 15%, that means the recombination frequency between A and B would be how much? 21 minus 15, that is 6%. So if I can say that the gene looks, up till now what I could understand is that the genes look, arey yeah, just a minute, huh? Hmm. That the genes look something like this: A, B, and C. This total is 21. This is 15%. It is given in percent. So, I mean, you can calculate it in centimorgan. And this is 6. Now, from this, what can be inferred? The gene order is A, B, and C. Okay, A, B, and C. And the distance between A and B is 6 centimorgan. Okay, this looks fine to me. Okay, chalo. The gene order of the order of the genes is A, B, and C. And the recombination frequency between A and C is 21%. Recombination frequency they are asking. Okay. The order of the genes is ACB, CB. It doesn't look really nice to me because the distance between the two extremes is 21, and if A and C and B are 15%, then it would be 6 only. So this doesn't look right to me. The order of the gene is ACB and the recombination frequency ACB, and the recombination frequency between A and B is 21%. It doesn't seem very correct to me. Okay. The the order of the gene is CAB and the recombination frequency between A and B is 6%. This can be true because either it can be ABC or C. Okay, CAB they are saying. Huh? It can be, if I am writing it as CAB, then recombination frequency between A and B. How, what is given here? The recombination frequency between B and C is 15%. If this is 15%, I don't think this should be the correct answer. So next is also there. The order of the gene is BAC and the recombination frequency between B and A is 6%. Between B and A. B and A is 6%. Hmm.
That's why I written, madam, ki A cannot be in between and C and B cannot be at the extreme.
Hmm. Now let us see what are the correct answers. The order of the gene is A, B, and C, and the distance between A and C is 6 centimorgan. Okay. This is what we have said, it is correct. The order of the gene is ABC and the recombination frequency between A and C is 21%. This also we said correctly only. We said this option is incorrect. They are saying the order of the gene is ACB and the distance between A and C is 6 centimorgan. Let us again see this statement, why I chose it as incorrect. The order of the gene is ACB. If it is A, C and B, it should be 21, and the recombination frequency between C and B is 15%, and the distance between A and 6. Yes, this is also correct. This can also be possible. See, if it is A, C, B also, then also it is possible, because here they have given that the recombination frequency between C and B is 15%. So if C and B is 15%, then obviously it would be 6%. Okay. So this is correct. This is correct. Then the order of the gene is ACB and the recombination frequency between A and B is 21%. This is also correct. Yeah. So I'll just explain this question to you now. Okay. See, three genes are there, A, B, and C. As of now, we don't know what is the order of the genes. We just know that the value of the two extremities, that means the distance between the two extremes is 21 centimorgan. That means three genes are there. So the distance between the first gene and the third gene, that distance is 21 centimorgan. Okay. We don't know which is the first gene and which is the third gene as of now. Fine. Then what are they telling? They are saying that the recombination frequency between C and B is 15%. This we know that B and C is 15%. B and C, it is 15%. So obviously it would be 6% or 6 centimorgan. So A and B would be 6. So this is 6% or in centimorgan, whatever. So this can be one of the way in which it can join. Or what else can be the way? Just remember that we have to keep B and A together, and A and C like this apart. So if you can see here, if I take it as ACB, if I write it as ACB, then can it be correct? Yes. Why? Because B and C is given as 15%. Just a minute. I think I put the wrong option. Ha. See here. ACB. A and C. Ha. Just a minute. Ha. B and C is given 15%. B and C is given 15%. That means A and C would be 6%, and this total would be 21%. So any of the way the genes can be arranged. Here, any of the ways gene can be arranged. So the correct answer would be A, B, C, and D. These would be the correct answers. Okay. I hope this is clear to you. If you have problem, you can ask me again. Yes, Ajay, you can speak.
Madam, I think it cannot be arranged as CAB. A cannot be in between.
So this that's what I said, na, that E is not the correct answer.
Okay, okay.
E
Thank you.
cannot be the correct answer. You see, it is CAB, na. If I write it as CAB, and here they are saying in this option the order of the gene is CAB and the recombination frequency between A and B is 6%. It cannot be, because these are the two extremes. If these are the two extreme, then this should be 21, and they have already given in the question that B and C is 15. So if B and C are 15, then how can CAB be the answer? So CAB cannot be the answer. So B and C they cannot go on extreme, because it is already given that B and C, the recombination frequency is 15%. So they cannot go on the extreme points. So this is also incorrect, this is also incorrect. Okay, I hope it is clear to others also. Okay, next question is, so after this question, I'll do is, like I told in the last class also, that after, as soon as we finish all the questions, we'll just quickly try to summarize whatever has been whatever topics or whatever course has been covered. Okay, so because there are other important uh, two three things that I think should be discussed, just we'll try to summarize the all the three classes of this week 3. Okay, after solving this question, whatever time we are left with, we'll try to solve those, I mean see those things. Okay, so this question says, just a minute. Hmm. Which of the following statement is or are correct regarding meiosis and recombination? A non-disjunction event during meiosis 1 has a higher probability of producing a progeny with a defect in the ploidy number. A non-disjunction event during meiosis 2 has a higher probability of producing a progeny with defect in the ploidy number. Complete linkage allows gene to have a recombination frequency of 50%. Complete linkage allows gene to have a recombination frequency of 100%. For completely linked genes, 100% of the offsprings will have a parental genotype. For completely linked genes, 50% of the offsprings will have parental genotype. 3 minutes for this question, and then I'll switch the screen. Ah, sorry. I hope you haven't saw the answer. Hmm. Try to solve this question. 3 minutes. Think and answer. One more minute left. One more minute. Now I'm switching the screen. Hmm. Let's see. Okay. So Ajay has answered A and E. A and E. Okay. And Chauvin has also answered A and E. And I think it is correct. Let us see. Uh, yes. So it is correct. So the correct answer is A, that is a non-disjunction event during meiosis 1 has a higher probability of producing a progeny with a defect in the ploidy number, and for completely linked genes 100% of the offspring will have parental genotype. Now there is one important concept that we should understand. So whenever like we dealt with 2 3 questions where we were trying to uh, uh, find out the correct gene order. So we are mapping the genes. Okay. So can anybody tell me this mapping is what kind of mapping? Is it a physical mapping or a genetic mapping? Can anybody tell me? Is it a physical mapping or a genetic mapping?
Genetic mapping, madam. The
Genetic
cells
mapping.
which are present, what the fate they will have. That
So what
means
is
they will
physical
live,
mapping then?
form, or they will die.
What is physical mapping then?
Physical mapping is just number of cells they have produced and the number of cells they remain. Physical mapping.
Physical mapping is basically calculated by the number of base pairs. Okay.
Yes.
And here we are talking about genetic mapping. So in genetic mapping we are trying to uh, uh, mm, and identify the distance between two genes on a chromosome. Okay, but it is not a physical distance, it is a genetic, on a basis of genetic distance it is. Now for this mapping your recombination is important to understand how much recombination has occurred, to understand about recombination frequency. It is important because why? Because I have two groups, group A and group B. Okay, in group A the distance between the two genes on a chromosome is less. That means genes are close together. Okay, and another group in which genes are distant. Okay, these are not close. So what does this from here, what can you infer? We can say that when genes are close together then they are linked. That means the chances of recombination is less. So genes are close together means recombination frequency is less. Recombination frequency is less. Whereas when genes are very much apart from each other, then there are chances of recombination. So recombination frequency is more. Okay. So that means if the two genes are very close together, the chances of recombination is less. So if you are given recombination frequency, that values of recombination frequency can be used to determine whether the genes are close or not. Just like here we saw in this question what we saw, we were given recombination frequencies. So the recombination frequency between C and B is 15%, and the recombination frequency between C uh, A and C would be 6. That means these two genes are closer together. From this only we could infer na. So recombination frequency, it indicates the distance between the two genes on a chromosome. Recombination frequency can be used in your genetic mapping, because greater the recombination, more is the distance. Remember this. Greater is the recombination frequency between two genes, more is the distance. So in this way, the values of recombination frequency can be used to analyze or to calculate the distance between the two genes. Okay. Then another important concept is, how do we calculate recombination frequency? Okay, I talked about that recombination frequency is inversely proportional to the distance. If the recombination frequency is more, it is directly proportional to the distance. If the recombination frequency is more, the distance is also more. If the recombination frequency is less, the distance between the two genes is also less. Okay, that all is fine. But the question comes that how do we calculate recombination frequency? So how can we calculate recombination frequency? This is a formula. Recombination frequency is equals to your recombinants, total number of recombinants upon total offsprings into 100. This is important. This is how your recombination frequency is calculated. So total number of recombinants, total number of recombinants upon total offsprings into 100. Okay, you can note it down, this formula, because anyways it's just like a general formula, like the selected by total into 100. That is how we calculate frequency in general for anything, right? Now let us see this question. Here also it was asked to calculate the recombination frequency between P and R. So now you know the formula. You can say that for calculating the recombination frequency between P and R, we need to identify the recombinations that are happening in P and R upon total number of offsprings into 100. Okay, that is what we did. I'm just explaining it again so that you can now relate it even better way, that for calculating the recombination frequency between P and R, we need the classes in which recombination in P and R are occurring. And then we found out that these are the classes in which recombination between P and R was occurring upon total number of offsprings, that means total calculation into 100. So this is how we calculate the recombination frequency. Is it clear to everybody? Is there any doubt? Okay. Now we have, I think, 15 minutes with us. Now we'll just quickly try to see what are the topics that were covered in this week's lecture. So it will just help us to revise and it will help you to solve your assignment questions also. So we started with differential gene expression. In differential gene expression, here we studied about selective nuclear RNA processing. So I just explained that how your differential gene expression can be governed by RNA selection and differential splicing, which is also known as alternative splicing. Then these are just the examples. I'm not going in detail with each and every figure because I won't be able to cover all the three lectures. Okay, so these are just the different ways by which differential splicing can occur. Here also this is another example. So you can see this is a muscle, which can be your smooth muscle, or it can have stripes or striated muscles, or it can be a smooth muscles. So depending upon which axon is taken, it decides what kind of muscle would be formed. So you can see that for the stripes, for the striations, these two axons are responsible. So if these two axons are there, then they would be striated muscles. If these two axons are not there, then it would be smooth muscles. Then for myoblast, like this, it is responsible. For smooth muscle, this red one is responsible. So like this, you can see that the differential selection of which of the axon is taken, that decides what kind of muscle would be formed. This is just an example. This is again another example of alternative splicing. Okay, this is again example. Then so first is selective nuclear RNA processing, then selective mRNA translation. That means differential gene expression, because from after this class, differential gene expression topic would be finished. That's why we are again revising it. So differential gene expression can be done by different ways at different levels. Differential gene transcription, that means transcription of selective genes, and this differential gene transcription can be achieved by enhancer, promoters, silencers. Okay, so here your concept of enhancer, promoter, and silencer will come. Then selective nuclear RNA processing, here the concept of alternative splicing will come. Now we come to selective mRNA translation. That means the ultimate thing is how a gene would be expressed only when your mRNA is translated into a protein. If your mRNA is not translated into a protein, then will the protein be expressed? No, because the process is that first DNA is converted into mRNA, then mRNA is converted into protein, and when a protein is formed, then only we say that a gene is expressed. When I say that this gene is expressed in our body, that means the protein which is specific to that gene has formed. This is very basic general concept, you should understand this. What is the meaning of expression? How you can say it is expression? Does it glows? Does it emit some light? No. That means that the protein is getting produced, that gene is producing its protein. That is the meaning of expression. So selective mRNA translation means that if we want the differential expression of gene, we can select that this mRNA should be translated in these cells and this mRNA should not be translated in these cells. So like this also you can do. And here comes your concept of microRNAs. Because microRNAs can um, um, inhibit the translation process also. So many genes, suppose a gene X, okay, a gene X, it does not need to be expressed in lungs, let's say, but the mRNA is there. Then how just what can be one of the ways by which this gene is getting expressed? Because some microRNA which is specific to this particular gene is acting upon this mRNA, and it is repressing the transcription of that gene. That can also be one of the reasons, I mean, one of the ways. So here comes the concept of microRNAs. You can see here, this is the microRNA gene which is known as pri-miRNA. Then DROSHA comes over it and it cleaves it, it becomes pre-miRNA. Then this pre-miRNA, it leaves the nucleus, and in the cytoplasm, cytoplasmic processing happens, and then dicer acts upon it, and it gets unwind and it forms your mature RNA and a passenger RNA. Passenger RNA, this is the mature RNA. Now this mature RNA can perform uh, repression, and or it can cleave the mRNA. Okay, I think this was the important thing in this lecture. Then in lecture 8, from lecture 8, it started all the genetic. From lecture 8, so in this week, you have 3 lectures, okay? In week 3, now you have the course content of week 4 live also. So in week 3, you had 3 lectures. The first lecture was about differential gene expression. It completed the topic of differential gene expression. And from lecture 8, your another topic has started, which is your genetic approaches, that we some of the questions that we have already understood. So there is not much to explain. So Mendel had proposed the 2 laws, the which are the most famous laws and the most studied and acceptable up till a certain time. And then of course we got to know that there are other phenomena like epistasis and all that occurs. But anyways, Mendel was the first person who could understand how the inheritance has occurred. So he this was the just a genetic, the way he did the cross, and then he made laws. The first law was law of segregation and the other one was law of independent assortment. So can anybody tell me the difference between the 2 laws? What is the difference between law of segregation and law of independent assortment? Anybody? What is the difference between law of segregation and law of independent assortment? Okay, so law of segregation says that if a gene is encoded by two alleles, okay, one gene is encoded by two alleles, then during gametogenesis process, these are segregated independently. Okay, those genes are segregated independent of each other. Each gamete receives only a single allele. That means if there are two gametes and there are two alleles, okay, so it is not like that one gamete will get both the alleles and one gamete will not have anything. No, each allele will go to one of the gametes. So it is like this. Okay, and the law of independent assortment says that the allele or a gamete receives for one gene do not influence the allele received by another gene. Suppose if I have two traits, tall and color, let's say. So, and this gamete has to receive one gene from this height and one gene of color, let's say. So, according to the law of independent assortment, um, this gamete will receive any of the genes which is not influenced by this. That means CC will not influence the uptake of TT, and similarly TT will not influence the uptake of CC. Both the genes independently they get inherited by the gamete. That means one gene will not influence other gene during gametogenesis. This is what law of independent assortment says, that all the alleles of the genes, they are independently getting inherited into the gametes. That is your law of independent assortment. And then we dealt with some of the basic, this is all just the basic how F1 generation, P generation, and everything. Test cross. What is a test cross? A test cross means whatever your phenotype is there, that phenotype is crossed with the recessive one. Wild type recessive. That is your test cross. Okay? With the wild type recessive, if you are crossing, that is your test cross. And then you just see, these are the things that you have to practice. Now, hi, here it comes your the figure that I was trying to say. See here, if you have a anti blood group A, this would be your genotype. This can be your genotype. Either it can be IAIA or IAI. And the antibodies which would be present are anti-B. And the antigen that is present is A. So likewise, you can see this whole table. And these are just the crosses. You have to go through these crosses actually, because these crosses cannot be explained orally. You have to yourself make in the copy and practice it. This is about recombination frequency that I was talking about. See, if the total distance is 17%, this is 9%, this is 9.5%. That means the distance between B and CN is little bit lesser than the distance between CN and vestigial. So this is just the we have already dealt in detail about this. So I would advise everybody to just whatever questions that we have dealt today, try to solve more and more number of questions of such kinds, because then as much as you practice, then only your concepts will become even more clearer. Okay, so practice as much as you can. If you have any doubts, you can ask me in the next class, and my all the lectures are recorded and they are available um, um, in my YouTube channel, Apurva Soni. If you type Apurva Soni NPTEL PMRF, then you can get the recorded versions of the lectures, and also the presentations that I am teaching you. Those presentations are also available. You must have a folder shared with you where you have the links of the I mean the files, the online uploaded files. You have a link, you must have that link, you that folder must have been shared with you. So from there you can access the presentations, these presentations as well. Okay, so yeah, just a minute. Yes, Ajay. I know you are
Question number 7, how it comes to 0.
so you have explained something, no? So can you explain it what you wrote? You have wrote it twice or thrice, I think.
No, madam, I just multiplied it 16 by 16 into 16. So it was 1 upon 4096, which comes to 0.0024. So that's
Hmm.
why I written it as a 0 probability.
That is even that is what I'm also thinking, but I'm not sure that whether this is the correct way to solve this question or not. So I'll just go through the question once again. That's why I'm not touching this question right now, because this looks to me as the correct answer, but I'm as long as not I'm not 100% confirm, I cannot tell it from my end. So let me just confirm it. It's a request for you also, for Ajay, and other one is Shovan, other two have left. So you also go through it and just read, try to find such similar concept, Google it or any book, what can be the answer. But yes, this can be the reason, I mean, this can be the logic. But I don't know how, why they have just similarly given, they have not given in fractions. They should have given in fractions. That makes more sense, because it's genetics may, it's not, we cannot do like this. But okay, ठीक है, we'll go through this question in our next class. Time is also up now.
Madam,
Just
one
remind me
minute,
if.
please. Madam, 1 minute. What my concept of test cross is, I will tell you. So, whether it is
Okay.
correct or not. Suppose a
Mm-hmm.
person is having blood group A,
Hmm.
that is phenotype, means his genotype can be A0 or AA.
Hmm. Yes.
I'm removing that I. Either means blood group A, it can be A0 or AA. This
Hmm.
person, if crossed with blood group of O, that
Hmm.
blood group O will definitely have both OO. Means,
Hmm.
uh, so after test crossing, if one of the blood group comes to 00, that means this person is having blood group AO, and of all the 16 they come AA. That means this person is having blood group AA. That is
Hmm.
test cross according to me.
Okay, so see, whenever we are doing any kind of test crosses, it is important to have generations. You understand? Test cross is, in a very simple language. If you want to know what is a test cross, it is basically crossing your selected one with the parental recessive phenotype. The parental So parental phenotype can be a dominant phenotype and a recessive phenotype. But in test cross, you are specifically crossing the parental recessive phenotype. Okay, so in that way it would be even more clearer. Like for example, if you're doing in labs, if you're doing any mouse experiments, so suppose if you want to create any inducible system, any knockout inducible system, so and at that point of time, if you have created any knockout, then at that point of time you do some kind of test crosses to create that inducible knockout system. So this test cross is actually very helpful if you are dealing with animal based studies. In animal based, you can just go and search it also. I think when you search it through animal based studies also, then your concept will become even more clear. Okay, so I think that is all for today. We'll end the session here. Thank you everyone for joining, although there were very just few people, but anyways. Okay, then we'll meet in the next week. Bye.